Hello everyone and welcome. I'm excited to introduce today's presentation on transcriptomics. On this first slide, you'll see the main topic, which is all about understanding gene expression at the RNA level. We'll explore what transcriptomics is, why it's important, and how it helps us unlock biological insights. Over the next few minutes, I'll guide you through the key concepts and recent advancements in this fascinating field. Let's get started. This slide doesn't contain enough information. That's why we're moving to the next slide. What is transcriptomics? Well, it's a fascinating field that focuses on studying all the RNA molecules within a cell or a tissue at a specific moment in time. Essentially, it gives us a snapshot of gene activity, showing us which genes are turned on or off. This is incredibly important because it helps us understand how cells respond to different stimuli, environments, or even disease states. By analyzing transcriptomes, we can identify patterns and changes in gene expression that are linked to health or illness. This connects directly to our overall theme of understanding biological processes at a molecular level. So, in simple terms, transcriptomics is about exploring the functional output of our genes, what our genes are actually doing, rather than just looking at the genes themselves. This makes it a powerful tool for research, diagnostics, and developing targeted treatments. Transcriptomics is all about studying the complete set of RNA transcripts that a genome produces under specific conditions or within particular cell types. It's like taking a snapshot of which genes are active at a given time. By analyzing different types of RNA, such as messenger RNA, non-coding RNA, and others, we gain valuable insights into gene expression patterns, regulatory mechanisms, and the broader functional aspects of the genome. The real power of transcriptomics lies in the technological advances we've seen, especially with high-throughput methods like microarrays and next-generation RNA sequencing, or RNA-seq. These tools have truly revolutionized the field, allowing us to identify which genes are expressed differently across conditions, discover transcript variants, and map out complex gene networks. This technology helps us understand development, disease processes, and how organisms respond to their environment. It's a crucial piece in the puzzle of molecular biology because it bridges the gap between the static genome and the dynamic biological functions. In essence, transcriptomics gives us a functional readout of the genome, helping us see how genes are actually working in real time. So why is transcriptomics important? Well, transcriptomics allows us to understand which genes are active at any given time in a cell or tissue. This is crucial because gene activity can tell us a lot about how cells function, respond to their environment, or even how diseases develop. By analyzing the transcriptome, we're essentially getting a snapshot of gene expression, which helps us understand the underlying biological processes. This knowledge is fundamental for advancing personalized medicine, developing targeted therapies, and improving diagnostics. Connecting this to the overall theme, transcriptomics is a powerful tool that helps us unlock the complexities of biology, leading to better health outcomes and innovative solutions. Let's take a closer look at the applications of transcriptomics. This field is incredibly versatile and plays a vital role in many areas of biological research and medicine. One of the key applications is in understanding gene expression patterns across different tissues, developmental stages, or disease states. By analyzing the transcriptome, we can identify which genes are active and how their activity changes under various conditions. This information is crucial for discovering biomarkers for diseases, helping us diagnose and monitor health conditions more accurately. For example, specific gene expression signatures can indicate the presence of cancer or infectious diseases. Transcriptomics also supports drug development by revealing targets and understanding how drugs influence gene expression. This can lead to more effective and personalized treatments. Additionally, in agriculture, transcriptomics helps improve crop traits and resistance by studying plant gene expression responses to environmental stresses. Overall, these applications highlight how transcriptomics connects to our broader goal of understanding biology at a molecular level and transforming healthcare and other fields through this knowledge. Now, let's take a closer look at some of the key techniques used in transcriptomics. On this slide, we've got a table that highlights three main methods: microarrays, RNA-seq, and qRT-PCR. Starting with microarrays, this is a hybridization-based approach. It allows us to detect known transcripts by hybridizing labeled RNA to complementary DNA probes on a chip. It's been a foundational technique for a while because it's relatively straightforward and cost-effective, especially when we're interested in profiling many samples against known gene sets. Next, we have RNA-seq, which is a sequencing-based method. This technique not only detects known transcripts, but also reveals novel ones. It's incredibly powerful because it provides a comprehensive view of the transcriptome, capturing both the quantity and sequence information of transcripts. This has really revolutionized how we study gene expression and discover new transcripts. Finally, there's qRT-PCR, which stands for quantitative reverse transcription PCR. This is a highly sensitive and precise method used to validate and quantify transcript levels. After initial findings from broader techniques like microarrays or RNA-seq, we often turn to qRT-PCR to confirm specific gene expression changes with high accuracy. Connecting all these, each technique plays a vital role in transcriptomics research, from broad screening to detailed validation. Understanding their differences helps us choose the right tools for our specific research questions, ultimately advancing our insights into gene expression and function. Let's take a closer look at the transcriptomics workflow. This process is essential for understanding how gene expression varies across different conditions or samples. It all begins with sample collection, where we gather the biological material we want to analyze. Once we have our samples, the next step is to extract RNA, because RNA reflects the genes that are actively being expressed at the time of collection. After extraction, we convert this RNA into complementary DNA, or cDNA, which is more stable and suitable for sequencing. The sequencing step is where we generate vast amounts of data, capturing the sequences of all the expressed genes. Following sequencing, we perform data analysis, which involves aligning the reads to a reference genome or transcriptome and quantifying gene expression levels. This analysis provides us with valuable insights into gene activity patterns. Finally, we interpret the results in the context of our biological questions, identifying differentially expressed genes or pathways that may be involved in the condition we're studying. Overall, this workflow is fundamental for unveiling the molecular mechanisms underlying biological processes, and it connects seamlessly with our broader goal of understanding gene regulation at a systems level. This slide doesn't contain enough information. That's why we're moving to the next slide.